to start with a quiz. So this is not a real challenging quiz. It tests whether you remember things from kindergarten or not. So when you get the sheet of the paper, you'll see that we're going to use 5 food color dyes. Extraordinary food color dyes. And it turns out that all of the food color dyes are made from the 3 primary colors. You all know what they are, right? Red, yellow, and blue.
Thank you, ma'am.
So your quiz is to, in the hypothesis row, predict what colors are used to make each of the dye colors. So I see some puzzled looks. I get that when I teach 7th graders this as well. So I'll help you get started. So the first dye is blue, right? So in the block under where it says blue, that's hypothesis. What color do you think you would make blue from? Blue. Very good. Blue. And you should be able to see the pattern after that, right? Okay, now it turns out that one of the primary tools that's being used in biology today is what we call a pipetter. It looks like this. And we're going to be pipetting 3 microliters of food color dye into a little slot in a gel. Right? I don't need to tell you how much 3 microliters is. 7th graders don't know this, but I'm sure you do. So to pipette very small amounts, we need a precision instrument. And what this has is a little plunger in here. You need to learn how to do this because you're all going to do it. So you hold it like this, with your finger under the hook, and your thumb here. If you push down the thumb, you'll feel it stop. And what you're doing is you're pushing down a metal plunger that's inside and displacing a certain amount of air. And when you let it back up, it will suck in a precise amount. We'll set this, it's already set at 3 microliters. We use pipette tips as a way to keep from contaminating the pipetter and making sure that we don't cross-contaminate our samples. So you put a pipette tip on, and what you're going to do is you're going to take one of these tubes of food color dyes and open it up, put your pipette tip into the dye. Well, first you push it down in the air to release the air. Put it into the dye, let it up, and you'll have 3 microliters in your pipette tip. Okay. Now, the challenge is to load it into one of these wells, and the wells are just slightly bigger than the end of this pipette tip. They're wider, but they're narrow. They're little slots. So, the trick is to use 2 hands. One to stabilize the pipette, which is going to shake. Okay. But one of the things I've observed, by the way, is that the easiest people to train how to do this are 7th graders. And then, after that, it's high school students. And the hardest ones are teachers. Because 7th graders have no fear, right? But teachers are afraid they'll look stupid. So don't be afraid. You're just going to come up and you're going to just carefully load the dye into a slot. And then we'll run the electrophoresis. I'll talk some more while it's running, and then we'll analyze the results. So, everybody who's done eating, I'd like to come up to the front of the room now. And Tracy and I will load the first one. You can watch us, and then everybody gets to load one by one. shells are running. Does anybody have any questions so far about what we've been doing? All right, so this is, we do this as a practice in the lab that I teach for the students in the science lab. Students master the pipetting technique by running these gels, and then when it comes time to load their DNA, they usually do a very good job loading their DNA. So, how did I get into science? Well, it's sort of the opposite of the story I told you first, because when I was in middle school and then ninth grade biology, I had teachers that had a teach. And I remember, particularly in ninth grade biology, one project we had was we went and got pond water, and then we had microscopes, and we were to find as many different creatures as we could find in the microscope and draw pictures of them. And we couldn't go online because that didn't exist. Page through books to try to figure out what they were. And then later on in the year, there was some woods on the school grounds, and we went out and we measured a plot of woods that was 3 meters square, and we were to identify every living thing in that 3 meter square area. So those were really memorable experiences where we were outside doing things, exploring nature, finding out how life works. And so I didn't get turned off to science. In fact, I don't know how I knew this, but I decided when I was in high school that I was not going to apply to Johns Hopkins for college because that's where I was going to get my PhD. Interestingly enough, it was the only school that accepted me. But it worked. So I've been doing science for a long time. As you might guess, I've been eligible for retirement for a long time. I started thinking about retirement. I was feeling old, wondering what I would do if I retired, and thought about some different things I might like to do. And finally came to the conclusion that I liked what I was doing far better than anything I think I was doing in retirement, so I just started my retirement. So I have an active research program. I've studied a lot of things, as I said, I'm a geneticist. So I've done a variety of things. For a while, I was studying fish genetics. And we looked at striped bass, and we were partnering with DNR. It used to be a really great experience to go to the zoo and do field work. Get out there before dawn and watch the sunrise, listening to the howler monkeys in the zoo. All the creatures coming out on the river. Of course, there's no access to the river anymore. But the problem with striped bass in this state is they're too much fun to catch. And so fishermen in this state catch 60% of all the adult striped bass every year. Which, you can do the math, there's no fish after a few years. But to prevent that, DNR goes out and catches striped bass during spawning season. And they spawn them in the hatchery down at Monk's Corner, and then they stock all the lakes around the state with striped bass, so that there's always more. In fact, striped bass cannot reproduce in Lake Murray. So all the striped bass in Lake Murray are stocked. So in order for striped bass to reproduce, they have to spawn far enough upriver from the lake that their progeny will survive by the time they get to the lake. So striped bass eggs safe, at least in this part of the world, and the turbulence of the river keeps them in suspension. there's not a long enough stretch of river upstream of Lake Murray for that to happen. But a favorite place for striped bass to spawn is just below the I-77 bridge, right near the sewage treatment plant. There's a big sandbar for turns, and at the right night in April, there will be dozens of striped bass there spawning. And DNR thought that they're worried about the genetic health of the striped bass, and they asked us to do a study to try to figure out how the rapid reproduction worked in nature and to compare it to what they did. And they thought that there was group spawning, because you could go out there and you could see that there were fish everywhere and churning up the water, and eggs and sperm were being released. So the idea was everything was just being mixed. So what happens then is the eggs start floating down the river, and after 3 days they hatch, but the fish still have a big yolk sac, and they don't need to eat. But at 7 days they use up the yolk, and they need to eat. And if they're in a place where there's food for them to eat, they survive, and if they're not, they die. So there's a nice nursery area at the top of Lake Marion, and we went down there, and unlike that striped bass, the ones we caught were this big. They were only a few weeks old. And we isolated DNA from the fins of these fish. And we asked the question, are there full sieves? So are there striped bass that have the same mother, the same father? Now, let me give you some numbers so you can see how stupid an idea that was. So first of all, I just told you they thought there was group mating, right? So the chances of any fish having the same mother father were very small. Secondly, there's about 300,000 fish in the system who are sexually mature and ready to spawn. And we were sampling 300 fish. Guess what we found? No, lots of full sieves. We found lots of full sieves. So first of all, that tells us there's not group mating. And in the hatchery, you can see when they're spawning in the hatchery, a female getting ready to release eggs has a male right half a pace behind her and just tracking her. And as soon as she starts to release eggs, he releases sperm. I tell the 7th graders that when they mate, they don't touch each other. But apparently that's what's happening in the wild too. So that there's a male in the right place, and when those eggs come out, his sperm fertilizes all the eggs from that female. And another female will have another male. So there actually is pair mating going on. But we still shouldn't have found them. Because if you sample 300 individuals from 300,000 families, you're not going to find siblings, right? So why did we find them? Well, one of the years we sampled was 1992, and we discovered that 4 pairs of fish supplied about 25% of the whole year class. 4 pairs of fish. So these fish, you can see they get pretty big. That fish on the cooler there was a 20-pound. A fish that size can produce about 1 million eggs. And she releases them all at once. So they can make big families. And apparently what happens is sort of like buying lottery tickets. Right? You buy a lottery ticket, you scratch it off, you look at it, you throw it away. Right? Well, only rarely does someone hit a jackpot. Apparently, with the striped bass, that's what happens. You just have to be lucky that 7 days from whenever you spawn, your progeny will be in the right place, and the weather will be right, and there'll be food there for them to eat. Most of the time, none of your progeny survive. But it's not genetic, because next year, everybody buys the lottery tickets and different ones. So how did DNR react? Well, guess what they do? Out and they catch a few pairs of fish, they spawn them in the hatchery, they make sure their progeny survive, and then they spread them all over the state. They're doing exactly the same thing as the natural reproduction. So they were quite happy with our results, and that was a nice practical application of what's happening. Just making sure these are moving properly. So that we did striped bass projects for a while. We also developed some techniques for studying other kinds of fish. So we did work with swordfish. We worked with tuna. We were able to solve an international crisis that you never heard about because we solved it before it happened. So it turns out in the Mediterranean, they catch swordfish and they like them to be this big. American fishermen used to like to catch swordfish that were over 100 pounds. They grew back to less than 100 pounds. Although the swordfish stocks have been slowly depleted, and now they're happy if they catch one that's 100 pounds. And so they decided that since we know that swordfish come out of the Mediterranean into the Atlantic, that if they could persuade the Europeans to stop catching these little swordfish, then we could catch more in the U.S. coast. So we got samples from different places, and we were able to show that every body of water had a unique population, genetically unique population. And we could tell the Mediterranean fish from the Atlantic fish. And it turns out we were able to demonstrate that the Mediterranean fish just come out through the Straits of Gibraltar, and there's a nice feeding area right outside of Spain and Portugal there, Morocco. And the Atlantic fish know that too. They come there, they go together, and they all eat there. But when it's time to go home, they go home. The Mediterranean fish spawn in the Mediterranean, and the Atlantic fish spawn in the Gulf of Mexico. So there is no mixing, and the Europeans could stop catching swordfish altogether, and it would help our fishermen. Because the Mediterranean fish. So we relayed that to the State Department, and the whole issue was dropped. So much for helping. Mostly what I've done is work on bacteria. And so the bacteria.
That is.
So bacteria we work with are called Polybacter. Polys means stalk in Greek. So they're stalk bacteria. You can see the stalks here. And if you know anything about bacteria, you've probably heard about E. coli. They get bigger, they divide, they get two cells like starch. That's true for most bacteria. But these are different because when our stalk cell gets bigger, it splits in two, but one of them has a flagellum and allows it to swim, and the other one, the stalk, has a holdfast at the end, allows it to stick to the surface. And so the progeny cell swims away, and the stalk cell can go ahead and reproduce right away. And this progeny cell is actually an immature cell. It's incapable of replicating its chromosome and can't divide until it goes through a maturation process, loses its flagellum, and synthesizes a stalk. So this is a very interesting bacteria that has obviously cell cycle control of gene expression. And I naively thought that it might be simple enough that I could actually understand it. Having studied it now for 40 years, I can tell you it is much more complicated than I ever dreamed it would be, and certainly more complicated than one person could understand. But what we were able to show, we developed all the genetic techniques for this particular bacterium, and now there's about 40 or 50 labs around the world using this as a model system to figure out how the simple organism turns genes on their own. And we were actually able to come up with the idea, which has now been proven true, that they had to have some way to monitor where in this cycle they were. And we predicted the kinds of molecules they would use to do that. And it turns out to be the core idea about how to keep track of what you're doing and what you need to do next, how to turn on one set of genes and turn off when you're done and turn on the next set of genes. But now we're focused more on the actual DNA. This is a representation of the chromosome here with the genes being those horizontal blue bars. And you can't see it in this picture, but we can actually see it long enough to see the nucleotide sequence of these genes. We can do comparisons to figure out how genomes evolve. We're in the process of isolating these bacteria from the wild and sequencing their genomes. Something that I never dreamed we could do. I was involved in sequencing this genome back in 2002. One of the, probably about the fourth or fifth bacterial genome that was sequenced. We were involved with a $3 million grant to sequence one bacterium. Now I can do a bacterium for $1,200. So I can do things I never could do before. And we're also studying viruses that attack these bacteria. So this is an electron micrograph of one kind of virus, the most common kind of virus that attacks the bacteria. It's got this very long head and a flexible tail. And they actually have a little filament that comes off the top of their head and allows them to lasso the flagellum. And then the rotation of the flagellum allows them to slide down the flagellum so they get to the body of the cell. So here's the flagellum, they lasso it. When they get close, they swing around in this tail. They'll find a receptor close to the base of the flagellum and attach to it. So they have this tricky way of finding the bacteria, this long flagellum sticking out, and then sliding down the flagellum and attaching to it. And so we actually are isolating these viruses from the common in all water, naturally occurring water samples. So from ponds and streams and the river here. And we started isolating a number of these viruses, and then one of the undergraduates working in my lab said, why don't we get the bacteriology class to do it? And I said, great, why don't you write a lab paper? He did. And so we edited it together, we took it to the professor who was teaching the bacteriology labs, and he said, this is great, I was asked to introduce a research component. And so then we hired the undergraduate and one of my graduate students to teach the laboratory course. And we now have 48 students who have successfully isolated viruses this semester and are in the process of characterizing them. So it's a way that we can introduce research into the curriculum and have students really get engaged in authentic research. So those are some of the things that I do. One gel is done. So we're ready for the next phase of our experiment. So what I want you to do is come up and look at your gels. And I want you to make observations now about which colors you see in corresponding to which layer. And see if what you predicted is actually true. Come on up and look at the gels. So we had some good questions. One question was, why do the dyes move at a different pace? So I didn't give you the little spiel at the beginning. This is agarose gel electrophoresis. Agarose is a purified form of agar. Agar is a polysaccharide coming from seaweed. And it's a form of nice gel like Jello does, that has the advantage of it doesn't fall apart. Liquefied, stays nice and firm. So it provides a matrix with tiny little channels through it. And the electric current is going to pull the charged particles through it. And they'll be separated according to size, because the smaller they are, the more channels they can get through. Also by charge, so the bigger the charge, the faster they go. So that's what's separating these dyes. And some of you were surprised to find that there were extra colors that you didn't predict. And so the reason is that the dyes they actually use are not necessarily the color that consumers expect, and so you have to adjust the colors, just like mixing paints. They're mixing different amounts of different color dyes. And one person even noticed that there were two different color red dyes. Generally, when I do this with the students, I have them come up and make their observations. They only see the bright colored bands. And they report, and I have them come back and look, and tell them to look for the lighter colored bands. Because there's also another blue band in there that didn't run very far, and it's more of a bluish green color. So there's extra colors in there. And so part of the point of this is to show the importance of not only noticing the big things, but noticing the more subtle things as well. And oftentimes when we're doing science, it's something that's less obvious, which gives us the understanding we need to understand what's going on.